We are interested in if we can learn molecular features, for example, gene expression, from histopathology images. So we want to see if we can use deep learning approaches to predict gene expression and also do it in an interpretable way. First, we use convolutional neural networks to do cell type and tissue type classification on histopathology images based on pathologist annotation. We then take the output from convolutional neural networks to build a graph neural network model. Each node represents a cluster of cells that has a vector of features associated with it. Edges represent spatial information that connect neighboring nodes. Then we use this graph neural network models to predict gene expression. We then apply a method called GNN explainer to interpret our GNN models. GNN explainer picks out important node features as well as important edges that we can visualize on top of our histopathology images. So this approach is different from a lot of black box machine learning models. If we are able to visualize what are the important regions that we use to predict gene expression, and in general, GNN models predict gene expression with a pretty good accuracy. For most of the 12 genes, it achieved Pearson correlation of above 0.5. This tells us that the spatial information used by the GNN models are important to predict gene expression. We showed that we can accurately predict these 12 genes of TLS signature. What's more important is that when we stratify patients based on our predicted TLS gene signature, we found there was a significant difference in patient outcome. So we applied this GNN explainer on our GNN models predictive of TLS genes. We found out that the important edges picked by GNN explainer largely overlapped with previously known TLS, but extend beyond these regions. So this tells us that TLS might affect gene expression that are in the peripheral regions. And also we found out that features other than lymphocytes can be important for these gene expression prediction, for example, plasma cells. This provides us with a lot of space for hypothesis generation in terms of what regions of the slides or what biological features actually contribute to these gene expression. This is a proof of concept that we can predict gene expression using pathology images and also in an interpretable way. We envision this work can be expanded to additional clinically relevant transcriptomic signatures and therefore be informative for the choice of treatment strategies.